Hello, my name is Apurv Mishra,
I'm Nathan Solomon.
and we also have a third group member, Ahmed Shakil, who isn't here currently. We are conducting this project under the Topological Molecular Biology Laboratory at UC Davis, California, United States of America. Our project attempts to create a machine learning model that can predict how easily new mutations of the SARS-CoV-2 virus can infect a host.
To do this, we take an experimental model of the SARS-CoV-2 spike protein, that is, the protein used by the virus to infect a host, and we simulate mutations at two locations in the protein that are known to be most important for binding to the human host cell.
In order to train our machine learning model, we calculate biochemical properties for each of the mutated sequences, which the model will use to predict binding affinity.
To calculate binding affinity, we use third-party software to predict the three-dimensional structure of mutated proteins. We then computationally simulate docking the mutated proteins to the host cell to calculate their binding energy, which is a predictor of the actual virulence of the new SARS-CoV-2 mutations.
We employ multivariate linear regression, among other methods, to train our machine learning model for prediction of binding affinity. The accuracy of our model is given by various statistical outputs, such as the root mean squared error, or RMSE, and our model is trained to optimize this accuracy and be as effective as possible.
Our project is useful because the machine learning model will let us calculate the binding energy for a sequence without folding it and docking it to the human cells, which takes a long time. Being able to calculate binding energy several orders of magnitude faster makes it much easier to simulate future evolution of the virus. The model can also help find the biochemical properties that maximize binding energy, which would help us identify a worst-case scenario mutation. These methods will help scientists stay a step ahead of the virus's future evolution and potentially prepare vaccines for new strains before they appear in nature.
Thank you for your time.